In this video, we will discuss about this urocanase and cis-UCA presence depends on the bacteria class and microbiome. So, in the previous video, we have understood that some of the bacterium has this HUTC and HUTU genes that is responsible for converting cis-UCA using urocanase, because HUTC and HUTU genes can produce this urocanase that can help in metabolizing of cis-UCA, and thereby this process will be utilized for the bacterium growth. And this is the reason some of the bacterium survives or some of the bacterium increases with this cis-UCA treatment or UVP exposures, whereas some bacteria does not thrives or proliferate. And here some of the validations has been done using Western blot analysis of the urocanase present in two different bacteria that have been studied in the previous video also. So this is S. epidermidis and C. singulare, and they have checked the urocanase presence in the presence and absence of this cis-UCA and trans-UCA. So they check the urocanase in the cell culture media of those bacterium growth. It is S. epidermis and C. singularis. So what they have find out is that S. epidermis, which has this HUTC and HUTU genes presence, shows this urocanase presence in their cell culture media, whereas C. singularis, which does not have this HUTC and HUTU genes, does not show urocanase presence. And these things they have validated, quantified this Western blot and normalized it to the GAP-DH signals, and they are able to see that there is no significant difference between the urocanase 1 when this cis or trans UCA are not present compared to it with trans UCA presence or cis UCA presence. But when they compared these signals with this C. singulare, they are able to see a significant difference, and the urocanase 1 values is significantly lower in the C. singulare at their respective conditions, that is when no cis-UCA or trans-UCA presence compared with this no cis-UCA or trans-UCA presence in the S. epidermis and the other two conditions. Thereafter, they wanted to check whether this microbiome also has an impact on this cis-UCA presence in this S. epidermis bacteria. So in this case, they use the control condition. They use a disinfectant, or they clean the skin with this 10% chlorohexidimine, which will reduce the microbiome on the skin, and they expose these mice, that is the control mice and the disinfected mice, under UVP exposures and cis-UCA applications, and thereafter they collect the samples using adhesive tapes at different time intervals, that is 2, 4, 8, 24 and 48 hours, and then analyze the cis-UCA using HPLC MS. And then what they are able to find out is that in terms of this UVP exposures, they are able to see that only a slight significant increase in the disinfectant case of the cis-UCA levels compared to the non-disinfectant, and this significant difference continues for the 2 hours, 4 hours, 8 hours signals when the mice is exposed to the UVP conditions. However, when the mice is treated with cis-UCA, they are able to see changes only in the 2 hours conditions. After that, changes in the cis-UCA levels is non-significant. That can be seen here in the panel C of the right hand side figure. Together, both of these figure results indicates that for the figures on the left hand side indicates that is the urocanase presence in the different bacteria results in its proliferations when the skin is exposed to cis-UCA or UVP. When the skin is treated with cis-UCA or exposed to UVP, whereas When it compares to the microbiome, for the UVP exposure, it shows a significant difference. That is, for the disinfectant, it shows high cis-UCA levels compared to the non-disinfectant. But for the cis-UCA, they can see these significant changes only for this 2 hours after treatment with the cis-UCA and UVP exposure. So this in overall indicates the effect of the bacterial class on the mice growth rate, and also the effect of this microbiome on the mice growth rate through this cis-UCA changes. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that is provided in the description. Thank you for your time.